Hi children, last class we completed antigen, right? Now we are going to see about antibodies. When the antigen enter inside our body, what will happen? Our B cell differentiate into plasma cells. These plasma cells shoots the antibodies against the foreign antigen, right? Whenever the foreign antigen enter inside our body, at the time B cell differentiate into plasma cell. That plasma cell produces antibodies against the foreign antigen. Yes, what is antibody? See the point number 1, it is otherwise called as immunoglobulin. What is immunoglobulin? A globulin is a protein part. Antibody contain a protein part called globulin. That's what it is termed as immunoglobulin. Antibodies are protein molecule. When it is synthesized. Point number 3. It is synthesized when exposed to the foreign antigen. See the point number 4. They can combine with specifically foreign antigen. Antibodies can combine specifically with foreign antigen. When the foreign antigen enter inside our body, what will happen? Our body synthesizes B lymphocytes. That B lymphocytes produces an army of protein called antibodies. These antibodies fight against the foreign antigen. Yes, once again, when the pathogen enter inside our bodies, that time B lymphocytes are synthesized. That B lymphocytes produces an army of protein called antibodies. That antibodies fight against the antigen, foreign antigen. Point number 6. Antibodies are secreted in response to an antigen. When the antigen enter inside our body, what will happen? Our antibodies start to produce a response, immune response to a foreign antigen. Antibodies secreted in response to a foreign antigen. When the antigen enter inside our body, it triggers the antibodies. When the foreign antigen enter inside our body, it triggers the antibodies. Right? Point number 7. Secreted by the effect of B cell called plasma cell. Yes, B cell differentiated into plasma cell. That plasma cell produces an army of protein called antibodies. Yes. Point number 8. Antibodies are of basically 5 types. They are structurally, physiologically have different properties. Antibodies basically 5 types. Because different types of antigen enter inside our body, right? Different types of pathogens. Our body exposed to different types of pathogen. பலவிதமான கிருமிகள் நம்ம உடம்புக்குள்ள வரதுக்கு சான்சஸ் இருக்கு. அப்ப வரக்கூடிய ஆன்டிஜன் கேத்த மாதிரி நம்ம ஆன்டிபாடி என்ன ஆகும்? அதோட ஷேப் ஸ்ட்ரக்சர் எல்லாம் பேசிக்கா மாத்திட்டு இருக்கும். சோ அத பொறுத்து 5 டைப் ஆஃப் ஆன்டிபாடிஸ் பார்க்க போறோம். That antibodies are IgM, IgA, IgD, IgE, IgG. ஈஸியா ஞாபகம் வச்சுக்கணும்னா, ஈஸியா ரீகால் பண்ணனும்னா MAG. IgM stands for mu, IgA stands for alpha, IgD stands for delta, IgE stands for epsilon, IgG stands for gamma. Yes, in 1950, scientists named as Porter, Edelman revealed the basic structure of immunoglobulin. Yes, these are the 5 basic structure of immunoglobulin: IgG, IgE, IgD, IgA, IgM. It is not present in your book. Just for extra point I added here. These are the 5 types of immunoglobulins. See, if the immunoglobulin is simple, single, that is monomer. Then the immunoglobulin is dimer 1 and 2, which is linked. That is dimer. When the immunoglobulin have 1, 2, 3, 4, 5 structure, that is pentamer. Monomer, dimer, pentamer, based on the structure of antibodies. Monomer simple and single, dimer 2 antibody molecules, pentamer 5 molecules. It has 5 molecules structurally, right? IgD, IgE, IgG belongs to monomer. IgA belongs to dimer. IgM belongs to pentamer structurally. Yes. Now you are going to see about antibodies, type, location and function. As we studied earlier, 5 types: IgD, IgM, IgG, IgA, IgE. Right? Now IgD. IgD location always attached to B cell. IgM can be attached to B cell or otherwise they can freely float in blood plasma. Freely float in the blood plasma. IgG 75 to 85%. 75 to 80% of all antibodies in blood belongs to IgG type. IgA, it is present in the secretion of saliva, tears, intestinal juice and first milk called colostrum. After the childbirth, immediately the lactating breast produces first milk called colostrum, which is rich in IgA. That's what doctors advise young mother to feed the infant immediately because the first milk contains IgA antibodies. Yes, IgE present in the mucosal lining of respiratory tract, gastrointestinal tract and tonsil. Mucosa lymphoid tissue. Mucosa associated lymphoid tissue area contains IgE type of antibody. Now we are going to see about function of these antibodies. Yes, IgD function. It activates the B cell. IgM is largest antibody actually huge compared to the rest of the antibody. First immunoglobulin released during the time of primary response. IgG most abundant in blood plasma and then it has the capacity to cross the placental barrier. So IgG is passed immunity to the fetus. Yes, what is placenta? Placenta is a temporary part in the pregnant woman which transfer the nutrition to the fetus through the umbilical cord. Right? So, in the IgG antibodies ku என்ன பவர் இருக்கு? பிளசண்டாவை கிராஸ் பண்ணக்கூடிய பவர் இருக்கு. அது மூலமா இது யாருக்கு இம்யூனிட்டியை கொடுக்கும்? ஃபீட்டஸ்க்கு. ரைட், தட் இஸ் த characteristic feature IgG. IgA, secretory IgA because bars the body surface important for first defense. Secretory IgA because it bars the body surfaces and important for first line defense mechanism. IgE, troublemaker antibodies involved in allergies. IgE, it's important characteristic feature which involved in the allergic reaction. Yes, got it. Now we entered in your textual content basic structure of immunoglobulin. Basically Y-shaped. See, this is a Y-shaped structure and then it has 4 polypeptide chain. See to the picture, it has 4 polypeptide chain, 2 identical light chain. 2 identical light chain, 1, 2 and then 2 identical heavy chain. 2 identical heavy chain. Once again, light chain 1, 2. 2 identical light chain, then 2 identical heavy chain. So totally how many polypeptide chain it has? 4 polypeptide chain. Yes, 2 polypeptide chain are light chain, 2 polypeptide chain are heavy chain. In this identical light chain molecular weight 25,000 Daltons. 2 identical heavy chain molecular weight 50,000 Daltons. 2 identical light chain approximately it has 214 amino acid. 2 identical heavy chain approximately has 450 amino acids. Right? Number of amino acids in the light chain 214. Number of amino acids present in the heavy chain 450. Yes, polypeptide chains are linked together by disulfide bond. See, this is heavy chain, this is light chain. One light chain is attached to each heavy chain by sulfide bond. Sulfide bond. As such as 2 heavy chains attached each other with the help of the disulfide bond. Light chain and heavy chain, and then two heavy chains are attached with the help of the disulfide bond, SS bond. Once again I repeat, light chain, heavy chain is attached by sulfide bond, as such as these two heavy chains are attached by disulfide bond. Attachment of L and H chain forms Y-shaped structure. This attachment of L and H chains forms Y-shaped structure. Hence, antibody represent H2L2. Two heavy chains, two light chains. So, two heavy chains, and then two light chains. Hence, antibody represent H2 and L2. See, based on the heavy chain, based on the H chain, it is classified into 5 types which we studied now only. Okay, gamma, alpha, mu, epsilon and delta. Right? Heavy chain, based on the heavy chain, structurally and physiologically differed. Next, light chain. Listen carefully. Light chain in human, light chain has 2 types. One is kappa, another one is a lambda. When you go to the deep, kappa chain is formed of chromosome 2. Chromosome 2 makes kappa chain. Chromosome 22 makes lambda chain. It is actually a neat portion. Yes, light chain ரெண்டு விதமா இருக்கும். ஒன்று காப்பா, இன்னொன்று லாம்டா. ரைட், எய்தர் காப்பா இல்லை லாம்டா, ரெண்டும் சேர்ந்து ஒரே லைட் செயினில் இருக்காது. Based on the heavy chain, 5 classes of antibodies. But light chainல ஒன்று காப்பா டைப் இருக்கணும், இல்லை லாம்டா டைப் இருக்கணும். Yes, see to the picture, point number 8. Heavy chain have flexible hinge region. This heavy chain, this is heavy chain, fully, right? This heavy chain have flexible hinge region. This is a flexible hinge region. This region, flexible hinge region, location approximately middle. எங்க இருக்கும் அந்த hinge region? Approximately middle. See to the point number 9. L and H chain has 2 terminal. L and H chain has 2 terminal. C terminal, it's a carboxyl terminal. N terminal is a amino terminal. Carboxyl terminal, amino terminal. L and H chain has 2 terminals. Okay, 2 terminals. Y ஓட டாப்ல இருக்கிறது என்ன டெர்மினல்? Y ஓட டாப்ல இருக்கிறது அமினோ டெர்மினல். Y ஓட டெயில்ல இருக்கிறது கார்பாக்சில் டெர்மினல். Then see to the point number 10. L and H chain has 2 region. Variable region and constant region. Variable region, this is variable region. This part is called a variable region, and after that constant region. L and H chain has 2 region up to this variable region. The Y-shaped part, top part is a variable region. Rest of the part is a constant region. Constant region is much larger region than the variable region. Yes, got it? இன்னொன்று இதில் இந்த மேலே இருக்கக்கூடிய Y-shaped part, this part we used to consider fragment of antigen binding site. As such as, see, this tail or stem part of the Y is called it as fragment of crystallized region. Fab region, FC region. This Fab region is an antigen binding site region. FC region is a fragment of crystallized region. This fragment of crystallized region, the stem part of the antibody, used to bind to the B cells. B cells la, B cell oda surface la ஒட்டுக்கிறதுக்கு இந்த fragment of crystallized region உதவுது. Yes, got it. Next, now we are going to discuss about, as we studied earlier, there are two type of region, variable region, constant region, right? Variable region, point number 1, antibodies responding different antigen. Antibodies, this is antibody, it respond different antigen, have very different V region. See, have different V region. That region is called it as variable region. But C region, same in all antibodies. B region, see this region. C region, see this C region. This C region, same in all antibodies. Yes, 5 types of antibodies we saw, right? Antibodies responding different antigen have different V region, variable region. But C region, same in all type of antibodies. That is point number 1. Point number 2, each arm of monomer antibody VH and VL chain, VH and VL chain combine and form antigen binding site. Antigen binding site. Each arm of monomer that is VH, VL combine and forms antigen binding site. That is point number 2. Point number 3, antigen binding site shaped to fit specific antigen determinant. இந்த ஆன்டிஜன் பைண்டிங் சைட் இருக்கு இல்லையா, இந்த ஆன்டிஜன் பைண்டிங் சைட்டை நம்ம என்ன சொல்லுவோம், பாரடோப்னு சொல்லலாம். லாஸ்ட் கிளாஸில் நான் சொல்லி கொடுத்தேன். A part of the antibody which used to connect with the epitope, that is called it as paratope. வெளியிலிருந்து வர்றது ஆன்டிஜன். இந்த ஆன்டிஜன் சி இந்த ஷேப்பில் இருந்தால் நம்மளோட ஆன்டிபாடி ஒய் ஷேப்டு ஆன்டிபாடி இந்த மாதிரி இருக்கும். புரிஞ்சுதா? ஸோ பேஸ்ட் ஆன் த ஆன்டிஜன் பைண்டிங் சைட் ஷேப்ட் டு ஃபிட் த ஆன்டிஜெனிக் டிட்டர்மினன்ட் சைட். இது ஆன்டிஜன், அந்த ஆன்டிஜனோட டிட்டர்மினன்ட் சைட்டுக்கு ஏற்ற மாதிரி. இப்போ இமேஜின் திஸ் இஸ் ஆன்டிஜன், that antigenic determinant site will be like this. Antigen binding site shaped, antigen binding site shaped to fit specific antigen determinant, right? That is point number 3. Point number 4, each antibody has 2 antigen binding site, 1, 2. Each antibody has 2 antigenic binding site. Now constant region. We are going to discuss about constant region. This region, constant region. C region, in small in all antibodies. Constant region, small in all antibodies. For stem of antibody monomer. It is actually a stem of antibody monomer. Determines antibody class. This region determines the antibody class. Function is common in all antibodies. இந்த கான்ஸ்டன்ட் ரீஜன் வந்து எல்லா ஆன்டிபாடிஸ்லையும் ஒரே மாதிரியான ஃபங்க்ஷனை தான் ஏற்படுத்தும். எஸ், புரியுதா? சி, திஸ் இஸ் த பேசிக் ஸ்ட்ரக்சர் ஆஃப் ஆன்டிபாடி. As I told you earlier, see, this is the antigen shape. This antigen is connected to the antigen binding site. ஸோ எப்படி எபிடோப் இருக்கோ, இல்லை எபிடோப் ஆன்டிஜெனிக் டிட்டர்மினன்ட் இருக்கோ, அதே மாதிரி இந்த ஆன்டிஜன் பைண்டிங் சைட்டோட ஷேப் இருக்கும். ஸ்பெசிஃபிசிட்டி, ஆன்டிஜெனிக் ஸ்பெசிஃபிசிட்டி, தட் இஸ் கால்ட் இட் அஸ் ஆன்டிஜெனிக் ஸ்பெசிஃபிசிட்டி. See, these are the things we completed, right? Based on the antigen binding site will be there. That is called it as antigen specificity. Yes, 5 basic functions of antibodies: neutralization, opsonization, complement, activation. Now we are going to discuss about 3 basic functions of antibody: neutralization, opsonization, complement, activation. Neutralization. What is neutralization? Antibodies prevent a virus or toxic protein from binding their target. இந்த வைரஸ் தன்னோட செல்லுக்குள்ள டார்கெட் பண்ணாமல் இருக்க இந்த ஆன்டிபாடி உதவும். தட் இஸ் கால்ட் இட் அஸ் நியூட்ரலைசேஷன். நியூட்ரல் பண்ணிடும். அந்த வைரஸ் ப்ரொடியூஸ் பண்ணக்கூடிய டாக்ஸிக், அந்த பாக்டீரியா ப்ரொடியூஸ் பண்ணக்கூடிய டாக்ஸிக்கை ஆன்டிபாடி வில் நியூட்ரலைஸ். ஆப்ஷனைசேஷன். வாட் இஸ் ஆப்ஷனைசேஷன்? ஒரு பேத்தோஜன் வந்ததுன்னா அதை சுற்றி இந்த ஆன்டிபாடி என்ன பண்ணோம், கோட் பண்ணிடும், கோட்டிங் ஓவர். அப்போ ஈஸியாக நம்ம பாடியில் இருக்கக்கூடிய மேக்ரோஃபேஜுக்கு ஒரு நியூட்ரோஃபிலுக்கு என்ன செய்யும், அது ஒரு ஃபாரின் ஆன்டிஜன் தெரிஞ்சு என்கல்ஃப் பண்ணிடும், ஃபேகோசைட்டோசிஸ். So that process is called it as opsonization. Opsonization is called it as coating over. Yes, next complement activation. Antibodies attach to the surface of a pathogen. Antibodies என்ன பண்ணும்? pathogen ஓட surfaceல ஒட்டிக்கிட்டு cell activate the complementary system. Pores ஏற்படுத்தி, அதாவது அந்த ஆன்டிஜனோட பாடியில் குட்டி குட்டி போர்ஸை ஏற்படுத்தி அது மூலமாக அந்த ஆன்டிஜனை அழிக்கும். These are the 5 basic functions of antibodies: neutralization, opsonization, complement, activation. Next class we are going to discuss about the antigen antibody interaction. Thank you children.